truly honored today to have Frank come and speak to us. Frank?
Well, thanks a lot, Kate, for the introduction. Congratulations to you and Lisa for bringing this group together. I think having this group all assembled in one meeting will be the first, I believe. I think we'll learn a lot from one another. I'm certainly looking forward to the next day and a half. I'm going to talk about really my experience in working on NF1 primarily, and our attempts to find drugs that block the RAS pathway, initially for cancer, but also some of these drugs might be effective in individuals suffering from NF1 and other syndromes that we'll be discussing today. So that's going to be the focus of my talk. The theme will be that it's been almost 20 years since we started understanding the molecular basis of these diseases, and only now really are we developing drugs that are actually being tested in the clinic. And although this has been a long road, I think the next era of research will be much more focused on clinical research and clinical targeted therapies which hit the right targets that affect these diseases. So that's, I think, the optimistic future, although I will say it's been a long time coming, and probably many of you in this room share the frustration at the time it's taken to develop new drugs to treat these disorders. This is my talk with the catchphrase, this is the beating heart of signal transduction, because the RAS protein is heart-shaped and is at the center of many of the pathways that are relevant to our discussions today, and I think it's also really a beautiful picture. I need to disclose some conflicts of interest. I still have founder shares in the company Onyx Pharmaceuticals, which I founded in 1992. We spun out of actually Chiron Corporation, which is a company just down the road, next exit on the freeway, moved up to a site in Richmond, which is further up the freeway. And at Onyx, we developed the drug sorafenib, or Nexavar, which is actually, you see that? Barely, is in clinical trials right now for patients suffering from NF1. It's also been approved already for kidney cancer and for liver cancer, and I'll try and mention some of the ways which we've learned from treating those diseases that might be relevant to the discussion today, but I do have a conflict of interest there. Also, I'm on the board of directors of Exelixis, a company down in South San Francisco, which is a company that has MEK inhibitors, RAF inhibitors, and a couple of PI kinase and mTOR inhibitors in clinical development in phase 2. And I hope that some of these compounds will eventually be tested in NF1 and some of the other disorders that we'll be discussing. So those are my conflicts of interest. My awareness of NF1 really came in 1990. I was working at Cetus Corporation on the RAS project, which I started in 1984, when it was clear that mutant RAS proteins play a direct causal role in cancer. And my group started working on the biochemistry of RAS proteins and how the mutant proteins differ from the wild-type proteins. And Meg Trahey in my lab discovered the activity which we call GAP. So we were working furiously on trying to understand how GAP activity regulated RAS and trying to clone and figure out how these proteins worked. And then one day I got a phone call from Ben Lewin, who some of you will recall was the editor and czar of the journal Cell. And he called me up and said, Frank, I've got a paper which looks interesting, I'd like you to review it. And it's on the disease NF1, and the gene's been cloned, and it looks as if it might be related to the gene that we're working on called GAP. So of course I agreed. Obviously when Ben Lewin calls up, whatever you are, you agreed in those days. I said, yeah, I'll be happy to review it. He said, well, there's a catch. It's already on the way to the printers. All you can do is veto it. And I'm going to fax it right now, and I want you to know this afternoon, is it good to go or not? So that was my assignment. That was the review process for this paper, which some of you may remember this process. But if you look at the dates here, the paper was received at Cell on 24th of July 1990 and appeared in the August the 10th issue on the newsprint. So that was the level of intensity and excitement about cloning the NF1 gene. And I guess the excitement was that rival groups, this is Ray White's paper, rival groups are on the same path, and Cell was determined to be the first to publish the paper. So this is probably the fastest paper ever published, and again it was in a really exciting era. So my group at Cetus, was then Cetus, featuring a number of stars here like Gideon Bolak, that I'm sure many of you know, who's in the audience today. Having reviewed the paper, we were ready to go as soon as the paper was published. And we quickly cloned and expressed the GAP-related domain of the NF1 gene and showed that it does actually work as a GAP and downregulates RAS, having affinity purified it from bacteria and expressed it in yeast. So this is a project which we've mobilized our resources in Ray White's group, but also at CETUS, and put together this paper, which was published, again, with a record turnaround. So we didn't know about the gene until late August, but we sent the paper off in October, and it was published in November. So again, this was a time in which there was tremendous excitement in the field, and we thought the fact that the NF1 gene regulates RAS, and therefore loss of NF1 upregulates RAS, really nails the basis of this disease, and that all the drug efforts that were going on in many institutions to find RAS inhibitors could be applied to treat this disease. And I believe it's true to say that there was a tremendous optimism in the community, especially those suffering from NF1 or NF1 families, that we'll rapidly have drugs based on this kind of understanding of the basis of the disease. And again, it's almost 20 years later, now they're coming, and I'll describe why it's been so slow, really, and some of the complications that we've run into in trying to develop therapies based on this simple understanding that NF1 is basically caused by a loss of a negative regulator of RAS, which hyperactivates the RAS protein and the RAS pathway. So now we know that the neurofibromin protein, as it's called, is a part of a superfamily of GTPase activating proteins, all of which have this GAP domain, which is a domain, again, which binds to RAS in the active state and kicks it to the off state by a mechanism I'll describe very briefly. The best known member of this family, the top one, barely visible to the naked eye. You can see that dot. Anybody have one of those big green things which... So the P120 RAS GAP is an abundant protein that we identified early on, as did the group headed up by Jay Gibbs, who's also in the audience. And there was another tremendous, tremendous race to purify and clone RAS GAP, P120 RAS GAP. We lost that race to the group at Merck, who put their resources on it and did a beautiful job purifying and cloning the protein. We came in a miserable distance. Thanks, Jay, for your effort. But it was a very competitive time. Anyway, the point is that this protein, P120 RAS GAP, is now well understood. It has a number of domains, SH2 domains particularly, which are known to interact with phosphotyrosines. Therefore, it was relatively simple to figure out how this protein functions. Okay. Thank you. So we know a lot about P120 RAS GAP, but we still really don't know much about neurofibromin, other than we know about the domain that regulates RAS. So neurofibromin is a gigantic protein from 2,800 amino acids, highly conserved between yeast and flies and humans, and yet most of the protein is of an unknown territory in terms of its function. So it's been very difficult to figure out what the protein does in cells other than regulate RAS, and that's been a big sort of question mark over the field. When we lose expression of the neurofibromin protein, are we losing other functions that might be important in the disease other than simply regulating RAS? One of the complications has been it's just been really hard to work on this protein because it's so damn big. Also, the antibodies haven't been good, and it's been hard to get full-length clones of the protein to actually work on. A refresher on how P120 RAS GAP works. When growth factors are activated, the Gram2-SOS complex is recruited to growth factor receptors and turns on RAS as an exchange factor and kicks RAS into the active state where it turns on downstream signaling. And then later on in this process, P120 GAP is recruited to the same receptors through a similar kind of interaction, and this turns RAS GTP back to the inactive state and thus turns it off again. So P120 GAP, most of the function of the protein appears to be a sort of sensor of activated receptors, which then downregulates RAS accordingly. That's the general principle by which we think these GAP proteins work. They recognize specific activated states of a cell and then tune RAS down to a level, with the correct level, according to the activation status. So we sort of assume that neurofibromin has a similar function. That is, it senses something in cells. And when that something becomes a critical level, it then downregulates RAS accordingly. Now, whatever it is probably in the plasma membrane, since that's where RAS is, although it's possible that neurofibromin is recruited to the plasma membrane in response to a signal. We just don't know what the signal is. And we're extremely interested in finding out what it is because it seems to be presumably some fundamental signal in many types of cells, since even yeast have neurofibromin lookalikes when they obviously don't have receptor tyrosine kinase signaling. So it's probably some fundamental aspect of cell physiology which neurofibromin recognizes and then tunes RAS GTP accordingly. And that's what we want to find out, and we have an active effort in the lab to try and analyze that sort of biochemically, led by Erica Bell, postdoc in the lab, who, taking the lead of Klaus Shevsek, who actually first told us it was possible to express full-length NF1 cDNA in cells, having changed every third base in the gene to make it possible to clone the cDNA and express it in bacteria. So the last decade, we've been stuck with a clone you just can't propagate in bacteria. So we've been unable to do all the standard signaling things one does with a clone to figure out how it works for the last decade. So we copied Klaus Shevsek's approach and had a company rework the whole cDNA so that it now can be expressed. Tremendous expense, as you might imagine. But now the clone works, and Erica has TAP tagged so we can do affinity purification, mass spec of proteins that bind to it, and flagged it and GFP tagged it so that we can express it in cells and see if it translocates and what it binds to, and do some biochemistry on the protein. And this shows that the expressed protein does indeed downregulate RAS GTP, so the full-length clones are active. So we really hope that we can sort of crack this problem. Now we can actually treat this like a regular sort of cDNA and actually do some biochemistry on the protein and understand how it works, and also make better antibodies against it. These reagents will be available to the whole research community as soon as we've got them sort of verified, and we'll put them out there. So hopefully this will help move things forward. For now, we're stuck with the observation only that it regulates RAS GTP by kicking RAS back to the inactive state. This complex for the gap domain I think this was actually from NF1, with RAS was solved by Shevsek and Fred Wittinghofer with help from my lab, including probably Gideon Bolag, who we expressed this fragment of the NF1 protein and purified it. And the way the protein interacts tells us how the gap activity works, because basically RAS in the GTP state has a very low intrinsic ability to hydrolyze GTP. But when GAP binds to it, it creates an active site, which then is able to kick RAS GTP to the off state. And this is GTP in the middle here, and the details of the active site are shown here, where the blue stuff there is sequences from GAP, and that is the arginine finger circled on the left, which becomes part of the active site, and the green stuff comes from RAS, and you can see that the blue and the green side chains come together with GTP and water to create an active site for GTP hydrolysis. So when GAP binds to RAS, it forms this complex, triggers the GTP hydrolysis, and turns the RAS protein off. So that's how it works, and that's how normally the neurofibromin protein regulates levels of RAS GTP. That's how P120 GAP downregulates RAS after growth factor signaling. Here's another view of the same thing. The GAP, and there's the active site, and there's GTP. I'm just showing this to make the point that the residues which we know about that are mutated frequently in human cancer and lock oncogenic RAS in the active state are residues that are around the GAP site, and the most famous, of course, being glycine 12. So mutations occur at glycine 12 in many human cancers. Maybe 30% of all human cancers change glycine 12 or similar sequences to, say, valine 12. This distorts the active site and prevents the arginine finger from GAP from triggering the off switch. And as a result, RAS proteins stay in the active states persistently, signaling downstream. And that is one of the elements responsible for probably 30% of all human cancers. So again, it's sort of in parallel to our efforts on NF1 and related syndromes, was a tremendous interest in the research and biopharma community in finding ways of turning RAS off, and specifically mutant RAS in the case of the cancers that are driven by mutant RAS. We screened a long time ago libraries of small molecules that might trick GAP into working on mutant RAS, and failed to find any compounds that did just that. And we sort of gave up and moved on downstream, as I'll describe. But I think, what I know, there's a renewed interest in the biopharma world on finding ways of finding drugs that will basically mimic GAP and work on mutant RAS to turn off the mutant RAS protein, because this is one of the untouched cancer targets that no one's really been able to get a handle on in terms of direct therapeutic intervention. So when we start moving downstream, now it gets complicated. This sort of partly excuses why it's been difficult to figure out how to turn off these pathways, because the RAS proteins themselves activate numerous downstream
effector pathways. So just upstream, you have a number of receptor signaling systems that feed into RAS. There may be dozens of these, different types of receptors, as well as different specific receptors. These all have the function of kicking RAS into the active GTP state. And then it's the job of the GAPs to come along and turn RAS down to the off state. And again, here's P120 gap and neurofibromin, but there are many others which tissue type specific or regulate RAS under specific circumstances, none of which we really understand in any molecular detail. But before GAPs turn RAS off, it has a chance to interact with a number of downstream effector pathways. And of these, the best characterized, the best understood is the RAF MAP kinase pathway, which I'm sure you're all familiar with. Also, RAS activates the PI3 kinase pathway by direct interaction with one of the PI3 kinase subunits. This interaction, I would say, is a little sort of wobbly relative to the RAS-RAF interaction, because PI kinase can be activated directly by receptors binding to PI kinase without having to go through RAS. And it's not so clear, I think, how much RAS contributes to activation of PI3 kinase in normal cells, because it's sort of complicated. But RAS definitely isn't required to activate RAF kinase and activate the MAP kinase pathway, which is central to this meeting. This aspect of the interaction, we think it's regulated by proteins called Sprouty and spreads, which are, again, negative regulators of RAS signaling downstream and work by different mechanisms to the GAPs. And it's not really clear exactly how they perform this function. Maybe this will come up later in the discussion from Eric Legius and his colleagues later in the meeting. So this is the main pathway that RAS definitely regulates. It probably has a role in PI kinase, it's not so clear. But RAS also activates other downstream effectors. RAS binds to a whole gaggle of proteins here in a GTP-dependent manner, any one of which could be relevant to NF1 or other diseases in which RAS is hyperactivated. But really, for the point of view of sort of simplicity and really focusing on what we know, we tend to focus more on RAF and PI3 kinase and are sort of waiting better genetic validation of these other RAS effector pathways before we can really sort of get our head around the contribution that these might make to these kinds of syndromes and to cancer. So just sort of make things a little simpler, we'll just strip out the gaps we don't understand and focus on neurofibromin really and figure out, sort of ask the question, when this gene is defective and this off switch is defective, RAS accumulates in a GTP-bound state and activates downstream effectors. Now, an obvious question again is which of these two effector pathways is most relevant to the disease? And this is academically interesting, but also important to understand from the point of view of developing therapies. Because, again, I'm sure you're all aware of the fact that there are a whole pipeline of small molecule drugs being developed which hit different aspects of both of these arms of the RAS effector pathways. I'll summarize some of these, particularly on the MAP kinase pathway, but there are probably 20 drugs in preclinical or clinical development which hit either PI3 kinase or mTOR or other parts of the PI3 kinase pathway as well. And I think it's really important to understand which of these pathways plays a major contribution to these syndromes to help us fine-tune which drugs are most likely to be effective in different aspects of the diseases. So in terms of syndromes which actually cause hyperactivation of RAS, obviously we think that that's the major feature of the NF1 disease, but there are other diseases which affect P120 gap, and of course you all know about Costello syndrome, in which RAS is actually locked in the GTP state through activating mutations in HRAS. And then downstream of that, we can look at the other syndromes and ask perhaps which of the phenotypes of these other syndromes would suggest that NF1 is a MAP kinase disease or a PI3 kinase disease, or both. One of the things which I hope to get out of listening to the presentations as we go through today and tomorrow. So in the sort of MAP kinase side, we have CFC, Noonan's, the NF1-like syndrome, which are extremely interesting in this respect, and also a leopard, which seem to be mostly syndromes affecting activating the MAP kinase axis from RAS, and then a number of other syndromes that affect the PI3 kinase axis. It's my understanding of the literature that the phenotypes associated with these syndromes are more like NF1 than are these. I'm not an expert in this area at all, but I'll be happy to have that discussion as we go along. But it looks like, from what I understand, that this would suggest that upregulation of RAS GTP through loss of NF1 most resembles the MAP kinase syndromes, and therefore one can conclude that it's mostly a MAP kinase disease. If someone wants to differ with that, I'll be happy to have that discussion. But I think it's approximately true, but not exactly true, most likely, because RAS does many other things. But it seems like that's true to a first approximation, and if you accept that, then you focus on diseases of the MAP kinase, drugs in the MAP kinase pathway as the most likely way of treating the disease. And we have a number of drugs that are either approved or in development that hit either RAF or MEK. Now, you might have thought that drugs that target RAF and MEK do more or less the same thing, since, as drawn here, they're just in a linear pathway. But actually, that's an oversimplification. It's probably not true, because both of these steps of activation of RAF and of MEK are extremely complicated at the biochemical level, and hitting RAF and MEK are actually different in quite profound ways, which weren't so obvious when this whole thing started. But we have some drugs I'll mention briefly. Sorafenib I already mentioned when we developed Adonix, which inhibits RAF kinase and other kinases that I'll describe. Novartis 265 has a similar kind of spectrum, although it's much more potent than sorafenib by a factor of maybe 100. Both of these drugs stabilize the inactive form of RAF kinase and hold it in the off state. Seemed like a good idea at the time, if that had been the idea. There are other drugs which actually work differently, and these are ATP competitive, which actually inhibit the active form of RAF kinase. And this turns out to be a very important distinction, which I think Gideon will probably bring out in more detail in his talk tomorrow, so maybe I'll skip on that. But they are important differences. In the MEK world, again, we have allosteric inhibitors of MEK and direct ATP competitors. These are also in early stages of clinical development. Very briefly, sorafenib inhibits RAF1, which is what it was screened against. Also inhibits BRAF, not so good against the activated allele of BRAF, which is locked in the active state. Remember, this drug binds to the inactive form of RAF kinase and therefore has a tough time inhibiting the activated allele, which is important in its clinical development. It also inhibits VEGF receptor 2 and c-KIT. And for those of you who know the NF1 field well, will recognize that these are potentially good targets for treating NF1 in addition to RAF itself, because many of the lesions associated with NF1 are highly vascularized and probably could benefit from treatment with anti-VEGF inhibitors, and also that from the work of Luis Parada, c-KIT activation of mast cells plays an important role in development of some of the lesions in NF1 as well. So you could say by sheer luck, sorafenib might be a good compound for treating NF1 because it hits several of the potential targets. And again, it is in clinical testing as we speak. Binds to the inactive state of the kinase. And it does actually inhibit activated BRAF in vivo. This is from a melanoma patient with V600E activated alleles of BRAF, and you see inhibition, at least in this one patient in this one time point. But sorafenib failed to have any impact on V600E driven melanomas in a clinical trial. And we now know, despite all kinds of theories as to why that might be, it's just not potent enough against that allele. I think that's the most likely interpretation. And we think that because Gideon here, who's in the audience, was one of the stars of the recent ASCO meeting in Orlando, where his group, Plexicon, showed that a more potent version, a more potent inhibitor of RAF kinase, which does inhibit the activated allele, does show dramatic clinical benefit in patients suffering from the activated, melanoma driven by the activated allele of BRAF. So this drug succeeded where sorafenib failed, and therefore one would most likely include this is just a better inhibitor of the V600E allele. Gideon, I think, will address that in more detail. So this is, I would hope, will barrel through approval and will be one more drug that can be used, hopefully tested, in patients suffering from some of the syndromes we're talking about in this meeting. So we're all very excited about that. However, sorafenib did show surprising clinical benefit